DNA, or deoxyribose nucleic acid, is a long molecule that contains our unique genetic code. DNA contains 4 basic building blocks, or what we call bases. There's cytosine, guanine, adenine, and thymine. DNA and RNA are nucleic acids consisting of long chains called polymers of chemical units called monomers, which are called nucleotides. One of the 2 strands of DNA is a polynucleotide, a nucleotide polymer, which is a chain. A nucleotide is composed of a nitrogenous base, 5-carbon sugar, and a phosphate group. As you can see down here, here is our nitrogenous base, which in this case is thymine, our sugar, which in this case, well, in all cases, is deoxyribose, and a phosphate group. The order, or the sequence of these bases, form the instructions of the genome. DNA has a unique double helix shape, which is like a twisted ladder, as you can see over here in this figure. It is simply a twisted ladder. The bases on one strand of the DNA pair with the bases on the other side of the DNA. These form the rungs on the ladder. So as you can see in this figure, the blue, which is adenine, pairs up with the red, which is thymine, and there's a hydrogen bond between these, and they're pairs, so they will pair together. There is cytosine on one side and guanine on the other side, and there's a hydrogen bond in the middle, which these two will pair together. Each pair, like I said, is joined together by hydrogen bonds. The nucleotides are joined to one another by a sugar-phosphate backbone. And as you can see down here, here is our sugar-phosphate backbone. RNA, ribonucleic acid, is unlike DNA in that it uses sugar ribose instead of deoxyribose in DNA, and has the nitrogenous base of uracil instead of thymine. So as you can see, this is uracil, and it is, you have all your regular nitrogenous bases, and then on this side you have thymine, which is not on the RNA side. And RNA is usually single-stranded, as you can see right here. And that is the basics of the DNA molecule.
And they are DNA polymerase, DNA helicase, and ligase. DNA helicase separates the strands of DNA, goes up from the bottom to the top, and separates them and takes them out of the double helix shape into the leading and the lagging strands. And then DNA polymerase synthesizes or puts together the nucleotides that make up DNA from the 3' to 5' end only, which is the leading strand. So it'll go up through here and add the nucleotides, various nucleotides. And then ligase is an enzyme that brings together, or ligates, the Okazaki fragments, which are newly synthesized DNA fragments, which result from the separation of the lagging strand, which runs from 5' to 3'. And so it'll go up through here and link together the Okazaki fragments.
In this video, we will be talking about replication of DNA. Now, I do apologize, I don't have the same template my partners used. I don't have access to that right now. So I printed one off, and hopefully we will get the same effect. I'll explain it very thoroughly. It'll be very understood. It'll be a great thing. But I do apologize for that. DNA replication follows a semi-conservative model, which means half will be old and half will be new. And as we go on, I'll explain that in a little more so it's not as confusing, but you'll understand. So, the 2 DNA strands separate, as you can see right here. We like to call that a replication fork. DNA helicase, right here, is our enzyme that unwinds the DNA strands. So as if it pushes it apart to form 2 strands ready for replication. Each strand then becomes a template for the assembly of the complementary strand from a supply of free nucleotides. So here are our old strands, because it's coming from our original DNA, and free nucleotides will be forming the new strands. and I'll explain why. So each DNA helix has one old strand and one new strand, like I just showed. Here's our old, these two are our new. DNA replication occurs in the 5' to 3' direction. Therefore, the 5' to 3' strand is our leading strand. So here we go. This is where DNA replication occurs. It occurs in the 5' to 3' direction. As you can see, it follows the arrow. Even down here, 5' to 3'. Now this is a little different down here, but I'll come back and explain that. And like I said, therefore, the 5' to 3' strand is the leading strand. So here we have our leading strand. It is our 5' to 3' up here. Replication is continuous on the leading strand. So that's why we have continuous written right here, which means it's just going to follow right through. There's going to be no chunks, it's just going to smoothly go through there. DNA polymerases add nucleotides only to the 3' end of the strand, never to the 5' end prime. So here's our DNA polymerase, and here's the 3' strand. It's just going to add right on. It's never going to start in the 5' end. See, it can't get through there. And then here we have a whole different story going on. But right up here, it's just going to go smoothly through. Replication is discontinuous on the lagging strand, forming short Okazaki fragments. So here's our lagging strand. This starts at 3' and goes to 5'. It is our lagging strand, and here's our Okazaki fragments. This is discontinuous. As you can see, it's like chunked up. We'll explain why. It is because, like we said earlier, DNA polymerase can't start on this 5' end. It can only start on a 3' end. So therefore, DNA polymerase, does a little, then goes back, does a little more, goes back, does a little more. It can't go smooth and continuous in this way. It can only, do the start at the 3', which our 3' is way over here. So, an enzyme called DNA ligase links the pieces together and to form a single strand. So DNA ligase is going to come in and do its magic and connect these together. So it'll be smooth and continuous like the one up here. DNA polymerases and DNA ligase also repair DNA damaged by harmful radiation and toxic chemicals. DNA replication ensures that all somatic cells in the multicellular organism carry the same genetic information. So this is how replication takes place, and it will form a identical DNA. It will form identical DNA so that all the somatic cells in the organism that this is from will be the same. And that is DNA replication.
Okay, we're going to start with initiation, which AUG codes for methionine, which is when RNA polymerase is going to attach to the promoter, and it's going to go up, and it's called elongation, and it's going to separate, and then it's also going to add in free nucleotides. And then we're going to reach termination, and that's when the molecule reattaches, and it may be RNA.
In the translation process, there are 6 basic steps. During this process at the beginning, the mRNA grabs onto the ribosome and attaches itself. After it's attached to the ribosome, tRNA attaches itself to free the amino acids into what's called the cytoplasmic area, where all the amino acids are stored. Once the tRNA has them freed, it takes certain amino acids to the ribosome. The 4th step is that it's going to take place. The tRNA is going to deliver its certain amino acids that they got based on complementary pairing of the anticodon and the codon of mRNA. The enzyme will then hook on to the end of the chain. This will create a polypeptide bond. Finally, the protein chain will continue to grow when the tRNA keeps delivering amino acids to the chain.
They loved you, oh love, they don't love you like I love you. Oh down, they don't love you like I love you. Something don't feel right because it ain't right, especially coming up.